So our next speaker, as we transition, is Julie Keister from the University of Washington, who's going to speak to us about North Pacific climate variability and zooplankton variability.
Okay, thank you all, and thank you, Andrew, for inviting me to be here. Can you hear me? Is this working? Yeah, somebody nodded? Yeah. Okay, thanks. I can barely hear myself. So yes, I'm going to talk today about North Pacific variability, and particularly the Northeast Pacific, where I do most of my research, including Oregon, Washington, and most recently, since moving to UW, Puget Sound. You can almost see it on the map here. It's a lovely place to do research, I must say. It's an inland sea that's connected to the Northern California Current, and it's been a very important site of research on zooplankton population dynamics and behaviors by Bruce Frost for years and years, and I'm trying to fill his shoes now. So in the Northeast Pacific, we have a lot of climate variability that takes the flavors of those around the global oceans. And I do want to pause and thank people who are absolutely unashamed about putting ridiculous pictures of themselves online so we can have things to put in our presentations. Yeah, white and nerdy, that's me. So, like I said, these natural climate variations in the Pacific have the same types of flavors as around the world. We have decadal cycles with long periods of many decades, for the most part. Periodic events that have shorter time scales of maybe a year or even just a few months to maybe 2 years, and are somewhat predictable in time. And then we have interannual anomalies or extreme events that seem very unpredictable and have even shorter time scales of maybe just a part of a season to a year. So the observed responses that I'll be talking about are local changes in the zooplankton in abundance and biomass or species composition. And I'm not going to belabor this point too much in this talk, but abundance and biomass are really not a very good predictor or signal of climate variability, mostly because the different species can interact to either dampen biomass changes or accentuate them in ways that don't really relate clearly to climate. But species composition, at least in the Northeast Pacific, is a very tight indicator of climate variability. So that is what we have mostly been looking at over the past couple of decades. But I'll show you some abundance and biomass trends as well. The mechanisms in the Northeast Pacific, there's this intersection of lots of different water masses. So advection of communities in and out of a local area is a really important impact of climate variability. But within movement, there's also behavioral movement of zooplankton as they behaviorally move up and down in the water column to avoid stressful layers. Say overly warm surface waters will cause them to move down, or hypoxic or low pH waters at depth may cause them to move up. And that has implications both for their advection as well as trophic interactions, predator-prey interactions. And then, of course, aside from movement, there's also changes in population structure driven by, say, increases in temperature and nutrients and plankton. And the one that's hardest to research at all is changes in predation and how that shaped communities. So certainly in the North Pacific, El Nino is our biggest climate signal. It's been known for hundreds of years because of its big impacts on fisheries. It's an expression at the equator, but it has big teleconnective impacts throughout the oceans and across the world. And these come in the form of changes in storm tracks towards the North Pacific, towards the Northeast Pacific, as well as advection via propagation of waves. So the equatorial signals cause big ecosystem changes. And here's an indication of the periodicity. We get something like an El Nino every 3 to 6 years. Some of the biggest ones, 82, 83, 97, 98, and excuse my very lazy way of updating somebody else's lovely figure. I just pasted in 15, 16. But these really big El Ninos occurred since zooplankton time series began in the North Pacific. So we're starting to get a pretty good idea of what the impact, at least of these big El Ninos, is on the zooplankton. So, yeah, I already said a bit of this, but El Nino obviously causes warming, and that comes with changes in stratification, nutrient delivery, and then therefore you should assume some bottom-up ecosystem processes on the plankton. The wave propagation and advective pathways are very important in the Northeast Pacific, as are these changes in storm tracks, which change mixing of the ocean as well as precipitation patterns. So the strongest evidence, though, is this advective pathways. So these zooplankton in the Northeast Pacific are very subject to relatively small changes in water mass patterns. But during El Nino, there's really big differences. And the first sign of the El Nino changes is usually in the popular press when somebody finds a tropical fish on their fishing line or in their nets, and it gets in the paper, and we all know finally the taxa have arrived. So these are, we don't usually find marlins, but we did find a pompano in Puget Sound last year, which is just it blew everybody's minds. But we get these really odd guys moving north. But they're fish, they swim. That's not exactly an indicator of advection. It's an indicator that there's warmer water that they like to be in. A clearer example comes from the zooplankton, because during these strong El Ninos, we get offshore taxa that are hundreds of kilometers offshore, show up in our time series at the coast. And I think my pointer is dying. We get this Euphausid nyctiphonys simplex, which is a very coastal krill. It lives just on the continental shelf, typically south of Point Conception. And during strong El Ninos, it'll show up at Oregon. So that is a very clear sign of advection because they are not swimming up there. So we get this convergence of this warm water and southern community during strong El Ninos. Here's the time series of nyctiphonys. You can see it here. It really only shows up during the strong El Ninos. And just to make the point that the bottom-up processes are not as strong as advection, this plot is showing the El Nino time scales. Here's the 97, 98 El Nino, and then several weaker El Ninos. Nitrate, chlorophyll. Thank you very much. Now, which one's the pointer? Chlorophyll, and this is biomass of the southern taxa of zooplankton that show up off Oregon. And what you see is during this strong El Nino, we did have depressed nitrate and depressed chlorophyll, as you'd hypothesize during an El Nino. But that's not so true for some of these other El Ninos. Here we have elevated nitrate, depressed nitrate, elevated chlorophyll. There's this mix of signals, particularly during these Central Pacific El Ninos, which are slightly different flavor of El Nino. But even during the 2012 Eastern Pacific El Nino, tiny little signal really, but elevated nutrients, depressed chlorophyll, southern critters. So the sign of the El Nino through the southern and warm water critters is quite a bit stronger than the bottom-up processes indicate. Ta-da, okay, great. As far as our long period cycles, obviously the PDO is the best known and has the highest impacts. So, now I don't know what to do. The warm phase of the PDO has an expression of very warm temperatures along the coast because a coastal scientist named it. Looks like it would be the cold phase, but it's the warm phase. So all through the California current, we have warm sea surface temperatures during the warm phase, and cold temperatures during the cool phase. And here is the time series. And you see that it used to have a pretty long cycle, but recently it's been much shorter period phasing. So there's been some shift in that. But in general, we do understand the impacts of the phases of the PDO on the ecosystem, very generally. During the warm phase, there's a strong Aleutian low, which sets up the warm phase, enhanced drier circulation, weaker upwelling, higher sea surface temperatures, higher stratification, and lower nutrients. So it's all this like lower productivity system overall, with some caveats that there's some mix of signal in the upwelling and the bottom-up processes. And during the cold phase of the PDO, we get a very productive system. We get stronger upwelling, colder temperatures, and lower stratification and higher nutrients. So looking at the effects on the zooplankton, this is a study I did quite a while ago now, but it holds over time. I just didn't update the figure. So this is basically a principal component. It's a non-metric multidimensional scaling axis scores time series from a single location, NH5, of the copepod community. So I call this the Copepod Community Index, and it is just the first axis of an NMS ordination plotted over time. And the strongest signal that you see is the seasonal cycle, but you also see some low frequency variability, and that is strongly related to the PDO. It's a little easier to see when I take the anomalies of the CCI. So then during the warm phases of the PDO, we have a very different community structure than during cold phases. And these are pretty persistent. They don't bounce around too much. It is warm phase, cold phase, warm phase, cold phase. And it indicates a very, very different community structure in the copepods. So of our extreme events, definitely the most extreme we've had since I've been in this business is the blob. This was a warm anomaly that set up in the North Pacific in 2013, and a lack of winter mixing in the winter of 2013 to 2014 really set the stage for this to be such a strong anomaly. So here in the North Pacific, you see the blob. This is January 2014. It's still quite cold along the coast in the California current. July 2014, in the summer, the blob is starting to expand. Son of blob has developed off of California. I didn't name these things. The Bering Sea is starting to be affected. But you see that in the summer of 2014, the northern California current was still upwelling, is still quite cold. So 2014 in our time series looks like kind of a normal year until late 2014 when the blob hit the coast. And it hit it hard and it hit it sudden because of changes in winds and downwelling, which affected that surface water right up to the coast. So suddenly we had to deal with the blob. So I'm going to show you a time series of temperatures from Ocean Station Papa. This is 2008 to 2016. So here's 2013, 2014. You see 2013 summer was already pretty warm, but what made the blob was that we didn't get the normal cooling. So here the light blue is climatology. So we ought to have been quite a bit cooler than we were during that winter. And again, it's because we didn't get the big storms and the big winter mixing that would cool the ocean down. So the ocean stayed really warm, which repeated in 2014-15. And we had these winter differences of 2 degrees C and summer differences of more than 3 degrees C. So this was a huge temperature signal out in the middle of the North Pacific. Now on the coast, we had a different pattern, but we did see the blob pretty clearly. Here we have some oscillating through 2013 and 2014. 2014, as the blob is developing offshore, we are still cool, as I already pointed out. During summer upwelling, that holds the blob offshore. So in 2014, we had a normal upwelling season. And here's where, bam, blob hits. In the mooring off of Newport, it took about 4 hours for the temperature to change about 4 degrees C. So it really was this edge of a water mass that had been sitting out there just waiting for downwelling to occur to just hit the coast. So there it is, hit the coast. And then it stayed quite warm. We had really strong upwelling winds in 2015, but a relatively weak response. It got a little bit cold, but not nearly as cold as usual. And then persistent warming through 2016. So what did it do to the ecosystem? Well, there's a lot of stuff in the popular press about extreme impacts. Very low euphoused biomass by Tracy Shaw and Bill Peterson's lab, which had big effects on the Cassin's auklets, which primarily feed on euphausids. There were massive die-offs, hundreds of thousands of dead birds throughout the California current. There were abandoned and starved sea lion pups. I'm sure you all heard about that in the news. That was really sad. There was also an unprecedented toxic Pseudonychia bloom. And this is an image during that bloom, and I'm told that it was practically a monoculture. It was a really, really unusual event. We had extremely low coho salmon returns, which in the NEP is a really big deal, and all kinds of unusual sightings of those tropical taxa showing up. So these huge, weird things were happening, and probably this bloom was the most negative of them. For the copepods, copepod species richness was off the chart. We thought that the 97-98 El Nino was off the chart until we plotted this. But here we had a very different species assemblage, species we had never seen before. 17 new copepod species observed on the shelf in 2014 and 15, many of them from the open ocean, some from the south, but mostly it seemed like this kind of transition zone in North Pacific current taxa, indicating that big advection of the warm water onto the shelf. And here is just to make the point that that species anomalies was all warm water copepods. The colder water copepod anomalies were depressed. So it was these warm water taxa. So now a little bit about Puget Sound, which is different than the California current, but connected. Advection of water masses is not nearly as important in the signal in Puget Sound. It's connected through a strait that has many sills, so we do get advection of oceanic taxa, but for the most part, I think of Puget Sound as being somewhat self-contained and a really good example of how population dynamics can change the copepod community. However, we're just starting to get zooplankton data from Puget Sound. We started in 2014, and it's ongoing so far. But what I thought was really interesting was, so the shading is total zooplankton abundance. This is 2014, which is our kind of normal year. And then 2015, which was heavily influenced by the blob, we had much higher abundances of zooplankton. Mostly driven by copepods, which is not surprising. Copepods tend to be 80 to 90% of the abundances everywhere. But the copepods did great. Everything did pretty great. There were very few taxa that did worse in 2015 than in 2014, except for one that is very relevant to this crowd, which is larvaceans. These guys are the ones that build these big mucus nets that they shed every now and then, and they're fast sinkers, so they contribute to carbon export. These guys had a pattern I don't understand yet. We always have a regular April peak in them. The peak was depressed in 2015 and 16, but we typically also have a spiky but pretty high number of larvaceans throughout the summer and early fall. But that component of the larvaceans was really depressed during the blob. And this is something that's got to have big implications for the carbon cycling in Puget Sound, and I don't really understand yet enough about larvation biology to understand why these changes occurred. Okay, so I've been talking a lot about these community shifts, but I haven't been telling you much about the implications besides that larvation. So what, okay, plenty of time. The big difference between these warm water and cold water copepods is in both their body size, but also their life history strategies and how many lipids they pack on. So this is an example of two different congeners of Calanus marshalli and Calanus pacificus. Calanus marshalli being associated with the upwelling system and cold water. It's a boreal taxa. It's found all the way up into the Bering Sea. Calanus pacificus is typically found offshore and down in California. It's dominant off California. So this is a warm water associated, cold water associated. You can see the lipid sac in this female Calanus marshalli, which is mostly missing in Calanus pacificus. So this high lipid load is a really fundamental difference in these copepods. So if we revisit this image of the PDO cycling, what we have is that during warm periods, we tend to have these smaller bodied and less lipid-rich copepods, and during cold periods, the larger and more lipid-rich, and the cycling through these periods. So that for a long time, it was a hypothesis that this was actually an important control on the system. Recently, we've been testing that by doing more careful study of the actual lipid composition and the fatty acids, which is even more important than just total lipids, because individual fatty acids are necessary for different physiological functions. So several studies now are showing that indeed these warm water taxa have lower lipids, which we kind of already knew, but they also have depressed EPA and EPA DHA. I think I have that backwards, do I? No, that's right. EPA to DHA ratios. And these are two indicators of copepod reproduction. They're really necessary for egg production, and they're also really necessary for the growth of juvenile fish. And so these community shifts have big implications for upper trophic levels as well. This is here that summer averaged NMS axis, or first principal component of the copepods, plotted against annual coho survival. And what you see is that when you have this warm community, you just can't get good survival of coho. Whereas if you have a cold community, there's a huge spread, but you can get some very high survival. So there is some trophic transfer of these communities up the food chain. There are lots of other differences among copepods. So these are all females from Puget Sound. They also are in the California current. You can see immediately there's this huge spread in body shape and body size. And that has other implications for carbon cycling. In particular, for this crowd, maybe the larger fecal pellets is of interest, because big copepods have big fecal pellets which sink really fast and contribute to carbon cycling. Whereas these little guys have smaller fecal pellets, some of which barely sink, and so they contribute a little bit more to the microbial loop in the surface waters. They also have a wide variety of trophic roles, ranging from pure carnivory to herbivory. Their trophic roles do not match well to their size. For instance, this little Oithona, he's pretty much a carnivore. He'll eat some plants sometimes, but he prefers microzooplankton. I guess I should call it she. These are females. Calanus mostly eats diatoms when available, but she'll eat microzooplankton too. And there are others that are more strictly herbivorous. I don't even have detritivores up here, but they're very important as well. So there's this huge mix of trophic roles in the zooplankton as well, which influences the biogeochemistry. So shifts in communities shift those roles, relative roles. And then lastly, the larger bodied zooplankton almost all do very big diel vertical migrations. and that's to avoid predators, but it's also, not coincidentally, or maybe coincidentally, important in the active carbon flux. So the larger zooplankton are feeding at the surface during the day and respiring and pooping at depth during the night, whereas the smaller ones are staying in the surface layer the whole time. So all of these changes in the community structure have implications for carbon cycling and predator-prey interactions and potentially big impacts on the ecosystem. I put this in just because it's my favorite slide about copepods and how we can't think about copepods as being a single trophic level. And this slide came from Hui. I don't know, I think it's Melissa Patrician. Is she in the audience? No? Do people know her? I love this picture. Tell her I love this picture. I use it in so many lectures about why you can't just put a copepod box. Here, this is a Euchaeta eating my favorite copepod, Calanus. So I have a grudge against Euchaetas, but I think this is just such a cool picture. So that's all I have to say about that. So in conclusion, big changes in zooplankton with climate variability. Signals of advection in the Northeast Pacific are really clear and really strong. But we know that there are also growth processes and population dynamics, predation, et cetera, that are changing these copepod communities in ways that are much harder to understand. I think our biggest remaining challenge that we're just starting to really work on is separating that advective signal from population growth dynamics. So that's it for now. We don't know what the future holds, but we have some ideas, we have hypotheses, we have predictions, and if it wasn't for the blob, I would have said we had some things really well figured out, and now I have no idea. So I thank my funders and my contributors.
Thank you very much. Time for a question or two, and then we'll break. Up on the left side here.
Hi there, Patty Matrai. I know that your time scales start in the late 90s, but there was a huge event in 1982. Could you comment as to how much more or less the 2014-15 is to what happened in 82 and others? The data exists.
Yeah. There was a big gap in sampling between the early 80s and the late 90s. So I didn't put those data on the slides because it makes it really messy. But the 82 El Nino did show the similar community changes and advection as the 97, 98. Yeah, it was very similar, and those data do exist, you're right.
Rubauji from Hui. Very interesting talk, Julie. I'm not surprised to see community structure shifted to warm water community when warm water hits.
Yeah, by definition.
I'm more curious about, what's a residual effect? What's a possible impact for the year after? Do you see any of those kind of impact?
A lingering effect, you're saying. Yes, they do recover pretty quickly. There are a couple of examples where they didn't, and one was the big 97, 98 El Nino, when the physical signal of the El Nino dissipated much faster than the biological signal. So we think it's because the alongshore advection had pushed warm water so far beyond Oregon, up even past Washington, that the relaxation of that community took a long time after the El Nino was over. So it could have still been advective, but there's also some evidence from Dave Mackes's old work where a species of acacia during a very warm event found a niche, basically, up on northern Queen Charlotte Island, where it was able to stay for a couple of years after the event. And so there are examples of where populations sort of invade and stay for a while before they disappear again. But most of the community changes go pretty tightly with the temperature and the PDO, indicating a pretty quick recovery.